Nearly every organism that is made up of many cells begins life as the single cell of a fertilized egg. That single cell divides over and over again until eventually an embryo is formed that is made up of trillions of cells of many different types. Over time, the embryo develops into a baby. And even at a very young age, some of the baby's cells begin to wear out. In fact, in a typical human being, every second of every day witnesses the death of about 50 million cells. Therefore, new cells must be constantly produced to replace old, dead, and damaged cells. For cells to reproduce themselves, whether in a developing embryo or in a fully grown adult organism, certain definite steps must be followed to assure that the new cells will contain exactly the same genetic material or genes that were originally present in the parent cell. And the essential process underlying the reproduction of cells is called mitosis. Mitosis is defined as the duplication and division of the nucleus of a cell with its chromosomes during cell reproduction. Scientists recognize 4 distinct stages of mitosis. First is prophase, second is metaphase, third is anaphase, and fourth is telophase. An average of about 6% of a cell's total lifespan is spent in these 4 stages of mitosis, while the other 94% of its life is spent in a stage that is not considered to be a part of mitosis, called interphase. Because the events that occur in interphase allow mitosis to take place, let us begin our examination of mitosis by taking a closer look at interphase cells. Interphase is defined as the period of a cell's life cycle between one mitosis and the next mitosis, the period of cell growth. As can be seen in this microscopic image of an onion root, some cells are dividing. The nuclei of most of the cells are round and intact. These are interphase cells. Average human cells, such as these that form connective tissue, spend about 19 hours in interphase and only between 50 to 90 minutes in mitosis and cell division. However, there is considerable variation among different types of cells as to how much of their life cycles are spent in interphase and in mitosis. For example, this human nerve cell rarely reproduces. Instead, it can remain in interphase for up to 60 years. Because nerve cells rarely reproduce, even to replace dead or damaged cells. Nerve damage, such as results from strokes or spinal cord injuries, is usually very serious. On the opposite end of the scale from nerve cells are cancer cells. These purple-stained cancer cells, which stand out clearly against a background of red blood cells, were taken from a leukemia victim. Cancer cells like these do not remain in the interphase state very long. Instead, mitosis and cell division continues at a furious pace. In fact, cancer can be thought of as cells that never rest, that divide over and over again, never remaining very long in interphase. Even though it is often thought of as a resting state between cell divisions, a lot is actually happening inside the nucleus throughout much of this stage of a cell's life cycle. During interphase, deoxyribonucleic acid, or DNA, the enormous molecule that forms the cell's genes and that holds all the cell's operating instructions, duplicates itself. Deep inside the nucleus of interphase cells, the 2 DNA strands unwind as new copies of the DNA are created. Scientists refer to this process of DNA duplication as replication, because exact replicas of the DNA are produced. The replication of a cell's DNA makes it possible for 2 complete sets of biological instructions to be sent into the new daughter cells. During mitosis, DNA is found within chromosomes, but chromosomes don't actually exist during interphase. Instead, DNA and protein form the grainy threads we see here called chromatin. The presence of chromatin is one indication a cell is in interphase, for only with the onset of mitosis does the chromatin mold itself into the distinct shapes of the chromosomes seen here. Scientists still do not understand the exact role that chromosomes play within the cell, but it seems likely that chromosomes are the best way to package DNA, and consequently genes, shipment into the newly forming cells. The chromosomes seen here, taken from the salivary gland of a fruit fly, provide a convenient material for scientific study because they are 10 times longer and 100 times thicker than normal chromosomes. Working with these cells, scientists have discovered that the dark and light bands we see here are the actual location of genes on the chromosomes. And their studies have also shown that when a particular gene on one of these chromosomes becomes active, the spot on the chromosome where that gene is located takes on a strange, puffed-out appearance. During mitosis, the appearance, movement, separation, and disappearance of chromosomes is of critical importance to the cell reproductive process. In fact, recognizing what is happening to the chromosomes, we can identify each of the 4 stages of mitosis. Now let us follow the events inside of a cell as it starts to undergo the intricate process of mitosis. The earliest sign that a cell is leaving a phase and is entering the first stage of mitosis, called prophase, is the chromatin begins to form itself into definite shapes of separate chromosomes. And at the same time, the nuclear membrane that separates the nucleus from the cytoplasm begins to be absorbed into the cell. As these things happen, the tiny nucleolus found in the nucleus also disappears. Thus, prophase can be defined as the stage of mitosis when the chromosomes first appear and the nuclear membrane and nucleolus disappear from view. As prophase progresses, the chromosomes become more distinct, and now that the chromosomes can be seen, a rather odd fact becomes apparent: that chromosomes are always found in groups of 2, called homologous chromosomes. This means there are 2 copies of each chromosome present in every cell of the body, with the exception of certain sex cells. For example, there is 1 pair of tall chromosomes and 1 pair of short chromosomes. This state, where double chromosomes are present, is called the diploid condition of a cell. The diploid condition of a cell means that there will be 2 complete sets of DNA instructions present inside its nucleus, so that if something is wrong with one set of instructions, the other set will still be able to provide the cell with the information it needs to function properly. Under close examination, we find that in early prophase, each individual chromosome, whether tall or short, has a rather thickish appearance. This is because the DNA of each chromosome has replicated itself during interphase. On looking even more closely, we see that each chromosome actually consists of 2 parts called sister chromatids that contain the duplicated DNA. And at first, sister chromatids are stuck very close together all along their lengths. It can now be seen that during prophase and part of interphase, there are actually 4 complete sets of DNA instructions present, because the DNA of each homologous pair is doubled at this point. The sister chromatids are held together at a specific region called the centromere. And together in this way, the chromosomes begin to arrange themselves so they can separate into the newly forming cells. And as they do so, the sister chromatids become much more obvious. Besides the changes in chromatin and chromosomes, other important events are also occurring inside the cell when prophase begins. In animal cells, a structure called a centriole or centrosome divides into 2 daughter centrioles that migrate to opposite ends of the cell. Between the centrioles, a delicate arrangement of microtubules called the spindle is formed. Microtubules that make up the spindle are called spindle fibers. Spindle fibers are critical to cell reproduction because they help arrange the chromosomes and later in mitosis separate them into 2 equal groups. In normal cells, All the complicated events that define prophase take between 30 and 60 minutes to complete. Finally ending when the next stage of mitosis, called metaphase, is reached. Metaphase, the second stage of mitosis, is defined as the stage of mitosis when all the chromosomes are lined up along the center or equator of the cell. Throughout this short 5 to 10 minute stage, the chromosomes are attached to the spindle fibers, and the centromeres that bind the sister chromatids together split apart. The splitting of the centromeres signals the start of the third stage of mitosis, called anaphase. Anaphase is defined as the stage of mitosis when the sister chromatids separate and move toward opposite poles of the cell. And when this happens, they are no longer called sister chromatids. They are now called daughter chromosomes. This movement happens fairly rapidly, in about 5 minutes, with the result that the spindle fibers disappear from view, and a full diploid set of chromosomes is now found at each end of the cell. The fourth and final stage of mitosis is called telophase. Telophase is defined as the stage of mitosis when the new daughter chromosomes change back into the threads of chromatin, and new nuclear membranes begin to form. Also during telophase, new nucleoli appear in each newly forming nucleus. As the final stage of mitosis concludes, the cytoplasm divides in half as cell membranes close up around the two new daughter cells. This final process of cell reproduction is called cytokinesis. Cytokinesis is defined simply as the division of the cytoplasm during mitosis. Between 10 and 15 minutes are required to finish both telophase and cytokinesis. When the entire cell reproductive process is completed, the two new cells are returned to the interphase state, and each possesses the identical genes of the parent cell. These two new half-size cells then go on to grow larger until each achieves full size of the original parent cell. The stage of mitosis seen here is called chromatin shortens and thickens to form these distinct structures called DNA is replicated during the stage between cell divisions called true or false. The division of the cytoplasm following mitosis is called cytokinesis. The first stage of mitosis, the stage when the nuclear membrane begins to be reabsorbed into the cell, is called In the first part of this program, we learned body cells reproduce themselves by mitosis, so that the new cells contain the same diploid or double number of chromosomes found in the parent cell. Now we will learn how certain diploid germ cells, found only in the ovaries and testes, undergo a process similar to mitosis called meiosis that results in the production of sex cells that have only half the number of chromosomes found in the parent cells. In the case of human beings, the diploid body cells all have 46 chromosomes, whereas the sex cells, the sperm and eggs, have only 23 chromosomes. This condition, where paired chromosomes are absent, is called the haploid condition of a cell. When two haploid cells, sperm and egg, join together at fertilization, a new diploid cell is formed that will then undergo mitosis over and over again, resulting in the creation of a brand new individual. Meiosis is similar to mitosis in many ways, but in other ways it is quite different. Now let us take a closer look at some of the details of this fascinating process. The earliest part of the first stage of meiosis, called prophase 1, begins just as it does in mitosis. Chromosomes form from chromatin, and these chromosomes contain DNA replicated during the preceding interphase. As prophase 1 progresses, two things happen in the germ cells that didn't occur during prophase in mitosis. First, the identical or homologous chromosomes form pairs with one another, so that the tall chromosomes find their mates, and the short chromosomes do likewise. This pairing of homologous chromosomes during the first prophase of meiosis is defined as synapsis. The second difference between prophase 1 of meiosis and the one we saw in mitosis is that parts of one paired chromosome can actually be exchanged with parts of its opposite pair. This process, called crossing over, is defined as the exchange of chromosomal material during synapsis and occurs only during the first prophase of meiosis. During the next stage of meiosis, called metaphase 1, the centromeres of the homologous chromosomes line up on the equator of the cell. In contrast, during mitosis, the centromeres of the homologous chromosomes align themselves in a completely independent fashion. During the third stage of meiosis, called anaphase 1, the homologous chromosomes, each made up of two sister chromatids, move apart, centromeres intact, one toward each mitotic center. In contrast, during mitosis, the centromeres separate during anaphase, and the sister chromatids move apart, new daughter chromosomes. During the fourth stage of meiosis, called telophase 1, each new cell contains just one of each homologous chromosome, and each of these chromosomes still consists of two sister chromatids. The fifth stage of meiosis, called prophase 2, follows right on the heels of telophase 1, without an interphase stage in between them, as happens in mitosis. This means the DNA of the chromosomes in the prophase 2 cells has not been replicated, and this explains why the chromosomes of prophase 2 cells have not sprouted any new sister chromatids. From now on out, meiosis follows the same stages as mitosis. During the sixth stage of meiosis, called metaphase 2, the chromosomes line up on the equator of the cell. And now the centromeres split apart. And during the seventh stage of meiosis, called anaphase 2, centromeres separate for the first time, and the sister chromatids become daughter chromosomes as they move toward the opposite poles of the cell. Finally, during the last stage of meiosis, called telophase 2, new nuclear membranes form. And as the chromosomes change back into chromatin, we can see that 4 new sex cells have been produced from the single original diploid germ cell. But since the DNA was only replicated once throughout a series of 2 cytoplasmic divisions, the final number of chromosomes in the sex cells is only half the number present in the diploid parent cell. In other words, the sex cells are haploid. Now, when 2 haploid sex cells, sperm and egg, join together, the fertilized egg will contain the same diploid number of chromosomes found in any body cell. Meiosis is very important. It enables all organisms that reproduce sexually, that is, require both male and female parents, to do so without continually multiplying the amount of DNA in the nucleus. For without meiosis, the human diploid number of 46 chromosomes reached 368 in just 3 generations, thus creating a very messy state inside the third generation cells. Meiosis also provides an effective mechanism whereby the DNA of the two parents may be resorted and recombined in new ways. And for this reason, no two human beings are exactly the same. Meiosis assures that each new generation will be unique and will always have new possibilities. The result is each generation will always have its own great athletes, scientists, artists, musicians, and inventors. And as strange as it seems, meiosis also provides a physical bridge between successive generations. And parents pass along a tiny amount of their own substance, their own DNA, to their children. As a result of meiosis, sex cells are formed that have the number of chromosomes of body cells. True or false? Pairs of identical chromosomes are also called homologous chromosomes. True or false? Normal body cells are diploid because they possess one of each different chromosome. True or false? During meiosis, the DNA is replicated twice. True or false? In plant and animal cells, meiosis occurs only in germ cells.